Thank you for the opportunity to talk today. I'm really happy, and I've seen so many great talks today on mussels and fish. My name's Matt Windle. I'm a research scientist at the River Institute in Cornwall, and I'm just going to be talking to you today about a program that I've been working on for the past 7 years in partnership with some great organizations and people, and lots of information to share. So I'll just dive into it. But the name of this presentation is the FINS project. So it's a large-scale study of the nearshore aquatic ecosystems and habitats of the St. Lawrence River. So just for context, the St. Lawrence River is the outflow of the Great Lakes, and I think we all know that. But just I love this image here, just showing that the largest freshwater system on the planet just flowing out of this pipeline at the end of the St. Lawrence River. And for context, the River Institute is located in Cornwall, Ontario. That's where the star is showing there, and it's a small nonprofit research institute that was started in the mid-1990s. And originally, it was started with the goal of looking at contamination in the Cornwall area of the St. Lawrence River from legacies from industrial waste, and it's grown in scope and employees and budget since that time, and to now including large-scale studies focused on fish, for example. And I've just highlighted the upper St. Lawrence River, this section of about 200 kilometers that starts from about Kingston, Ontario, and flows down to Montreal. That's primarily where I'm focused on today. So it's a big chunk of the river, and you can see this close-up view here. So we have lots of different towns on the Canadian side along there. I've also highlighted the indigenous areas of the river as well. And as I'm going to be talking about, the FINS project is a partnership project between the River Institute and the Akwesasne, Mohawk Council of Akwesasne, located in the same section of the river there. So the St. Lawrence River, it's, if you look at it on a map, it looks just like the big long blue line extending out of Lake Ontario, but it's actually, you can divide into these various components. So hopefully people have had a chance to visit Thousand Islands. It's a large section of the upper St. Lawrence River. It's over 1,000 islands actually. Lots of embayment, shallow areas. Moving downstream, you have the middle corridor, very few islands. It's much deeper, faster flow. And then you get into Lake St. Lawrence, which is this wider stretch that was artificially created by the construction of the Moses Saunders Dam in the 1950s that flooded upstream areas of the dam. And it's essentially an impoundment at this point that undergoes these really wide water level fluctuations. And then downstream of that, you have Lake St. Francis, which is located between the Moses Saunders Dam in Cornwall and the Beauharnois Dam at the end of Lake St. Francis. And this section has very, very small water level fluctuations. So lots of different things going on in the upper part of the St. Lawrence River, lots of different types of habitats, water level regimes, things to consider when you're looking at the ecosystems. And I love showing this instead of a static map. We use drones a lot in our research, and you can see it's a beautiful area. There's so much amazing habitat and scenery along the St. Lawrence River, and it's a massive river. One of the largest in the world, and you can see this is a Longsuit Parkway. This is 1,000 islands, lots of different types of habitats to examine. There's huge wetland complexes. This is around Akwesasne. This is a wide stretch in Lake St. Francis. Sorry, you can see the river widens significantly. So we visit all kinds of different cool habitats as part of this project. This is another wetland in Akwesasne. And the river overall is going through these massive changes. I just mentioned the construction of the Moses Saunders Dam in the 1960s really changed the flow of the river a lot, water level management. But climate change being this overarching concern. And then we have the impacts of invasive species, many of which were brought in during the post-construction of the St. Lawrence Seaway as part of bilge water in large ships, but lots of other ways that those species have entered the system as well. So the food web's undergone this really big, these big impacts, and we've seen some great presentations on the impacts of zebra mussels, quagga mussels, round gobies, things like that. The project I'm talking about today began in 2015, so a few years ago now. And the River Institute is this organization that was founded in joint partnership with the Mohawk Council of Akwesasne. So we've been working with the MCA on a variety of projects for the last decades. And just out of conversations, we were talking back in 2015, and we realized that there was this huge knowledge gap on the small fish populations of the area, the Akwesasne around Cornwall. People had mentioned that they weren't catching as many. We realized we didn't have a lot of great data on that. So we started this project to actually look at doing some fish survey, but also just incorporating and adding to that surveys of the habitats, trying to get a sense of how the ecosystem was doing, so looking at some indicators. And yeah, so that was the start of this great partnership between the River Institute and the Mohawk Council of Akwesasne, and we've worked together on this project since then. And shout out here to Abe Francis at the Mohawk Council of Akwesasne's Environment Program and other staff there that we've worked with over the years. So this project focuses on nearshore environments of the St. Lawrence River, and there are government agencies that have been doing decades of gillnet index surveys of the St. Lawrence River, and that's for larger fish, and it mainly focuses on sport fish, but larger adult fish size. There's not a lot of survey work done nearshore areas of the St. Lawrence River outside of the Thousand Islands, which is surveyed by in nearshore areas by Parks Canada in joint partnership with Muskies Canada. On the US side of the river, because there is a boundary that runs down the river on the US side, the New York side, there's been an incredible amount of work done by Dr. John Farrell's group at SUNY ESF in partnership with the government agencies there as well. But on the Canadian side, there's just this big knowledge gap outside the Thousand Islands on nearshore areas. So this project had a lot of objectives. I'm not going to read all these, but overall, the overall arching objective was to fill in these knowledge gaps on these nearshore fish populations. And we could build on this in so many ways with training and outreach and partnerships with other organizations. The survey involves using seine nets, and I've just put some of the details of that here. We do standardized seine hauls at a variety of different sites. We also, when we catch fish, we identify all them, but we also take measurements of length on all the fish too. And this survey runs between, sometimes it starts as early as April, but it typically runs between June and September. We come across, you can see a great photo here by Steph Hildebrand at the River Institute. But we come across a wide variety of species in the river, so we're documenting the abundance and species richness, as well as some of those morphometrics of the fish we catch. In addition, and the primary focus is on fish, but we're also documenting the aquatic habitats. We're looking at the substrates, the water depths, aquatic vegetation, and as part of that, we do a standardized vegetation survey at all of our sites as well. We're getting some great information on emergent and submerged aquatic vegetation communities at all the sites. And of course, this drives a lot of what we see in terms of the fish population. So it's really important that we document that carefully. We're measuring water quality, of course. So things like dissolved oxygen, temperature, and water clarity seem to be important factors for structuring fish communities. It's incredible how much of the shoreline of the St. Lawrence River has been modified by humans over the years. And so we're documenting that. There's not a lot of natural shoreline left. I mean, we're talking 200 kilometers of river, and it's quite rare that we actually find areas that have not been impacted. And thankfully, there's quite a bit of it left in Akwesasne. So that's one of our favorite areas to go to do our research. We've also started over the last few years incorporating the use of unmanned aerial vehicles, also known as remotely piloted vehicles or systems, or drones. So drones give us this great overhead view of our habitat sites, our survey sites, and then we can take hundreds of photos and produce overall maps, high resolution maps of our sites. And then we can get really good overhead look at the structure of the sites, and we can actually run the imagery through analyses as well, and through machine learning, categorize different elements of the habitat as well. And we've ground truthed this, of course, with some of the vegetation surveys. But we have some payloads on our drones, including multispectral cameras and thermal cameras. And through the use of multispectral cameras, you get views like this, where you can actually see algae growing on submerged logs and things like that. So we can tease out a lot more information from the multispectral imagery. And this is a growing part of this project that we're really keen on. In the last few years, we've also started the collection of environmental DNA at all of our sites. And this is just adding another tool to document diversity. So we do triplicate samples. We also have lots of controls in place. And then we filter our water samples on site. And then those are stored and frozen. And I'll talk a little bit more about who we're working with to look at that later in this presentation. So as part of this overall survey, we're documenting a lot of species at risk. We're filling in a lot of knowledge gaps on the distribution of these species. One of the main ones we focused on is pug-nosed shiner, but I've listed a few other here that we've really filled in. And again, this information is provided to government agencies and helps to contribute to different recovery strategies for these species. And of course, we're also documenting a lot of invasive species in the St. Lawrence River. Round goby is one in particular that seems to be found at almost all of our sites in the St. Lawrence River. We're also documenting a few new fish species moving in, including tube-nosed goby and tench. And you can see in the bottom left there some of our crew are goofing around in front of a massive Phragmites stand there, and that seems to be popping up a lot more on the river over the last few years. So that's the context of the project, where it's located, what the survey involves. I'm just going to go through now some project highlights. And again, really nice photo by Steph Hildebrand here, showing some of the fish diversity at some of our sites. Love those long-nosed gar juveniles. This map here, the series of maps, shows how the project has expanded over the years from 2015 to your left all the way to 2021 on your right. And last year we actually expanded into Lake Ontario. So we did a number of sites in Prince Edward County. And we're also moving up towards Montreal. So the project, just a overall message with this slide is that we're expanding our scope, geographic scope over the years. And after 7 years, we've had great success. We're getting close to about 160,000 fish documented as part of this project. It started from a simple question: how are minnows doing in Akwesasne? So we built a lot of data over the years. This is over 200 sites distributed from Prince Edward County now all the way downstream to Montreal. And this includes a massive amount of wetlands, different types of wide variety of habitats. We've documented 66 species since we started the project. I do have a little video coming up to show some of that diversity. And at a number of these sites, and really we got going with this last year, we've documented, we did 65 environmental DNA surveys as well. So that is being analyzed right now. A big part of this data set is also taking measurements. You can see a young of the year muskellunge there with the northern pike. So we take, and that helps us understand growth patterns of a lot of the fish we're seeing as well. Focusing in on Akwesasne, where the original project started, all these red points here are basically conveying the sites where we've conducted surveys over the years. And at a number of subset of these, we've actually visited every single year. So we're starting to build this long-term data set to look at changes over the years. And in 2017 and 2019, we had large flood years. So we were able to see some changes as part of this long-term monitoring now that's being set up. But yeah, over 110 sites now in Lake St. Francis and over 80,000 fish documented in this area. So we now have a fairly good understanding of the fish communities in this area and how they're changing over the period that we've been watching them. This is the pug-nosed shiner there. You can see its little itty bitty mouth. This is one of our priority target species. This is a species of risk. Then we've added over 20 new documented sites in the St. Lawrence River where they've been located now. And so this is a great highlight of this project, is just filling in these knowledge gaps for species of risk, and then we can show what types of habitats they are associated with, and really outline a path forward for protecting them. So I hope this is playing on your end. It looks great on mine, but we do come across a lot of really cool fish, lots of fish diversity. There's a tube-nosed goby group there and round gobies. As I mentioned, over 60 species have been documented as part of this project. And we've gotten to the point where we can actually go to various habitats and before we even put a net in the water, really predict what types of fish we're going to catch. As we should after visiting over 200 sites and documenting this number of species. And so lots of different species there. And as part of this, you can imagine this is a great platform for collaboration. We're covering a lot of the river. There's a lot of organizations and universities that are interested in other questions, ecosystem questions. And so we've been able to collaborate with a number of universities on the US side as well as Canada. I've listed a few here and I have a few slides to talk about those as well. But some really great collaborations just using this FINS project as this platform of collaboration. And if there's any interest in that, if there's people on listening in today or listening into this recording afterwards where you can think of a project idea that you'd maybe want to collaborate on, I'd love to hear from you on that as well. Our environmental DNA efforts have been largely helped by our wonderful collaboration with Dr. Steve Lahib's lab at Queen's University and his team of grad students. We've been providing our filter samples to them and PhD students like Stafford Miracle and postdoc Oriane Bernier. They're analyzing these samples right now, and I just saw some preliminary results from the metabarcoding last week where we're getting all these hits on different species that we've documented, physically caught them at our sites. And there's traces of these fish now showing up in the water samples as well, in addition to fish that we didn't catch at the sites. So we think this is a great new tool. I'd really like to move forward with this too as well, because when we do this kind of environmental monitoring, you get in the water and you do disturb fish. You disturb their habitat. You know, it can be a very temporary thing. But if there's a way that we can use tools more and more that reduce that disturbance, especially when we're looking at habitats for species at risk, that I think we should really prioritize that and move forward. This is a great project where we're able to combine all these fish catches with this environmental DNA data set now and pair those together and look at how well they're matching and complementing each other. So there's lots of results coming with that in 2022, this year. We're partnering as well with Muskies Canada and Parks Canada and Thousand Islands. And as I mentioned, they focus largely in that area. And we're contributing by covering a large area outside of the Thousand Islands for monitoring young of the year muskie. Their populations crashed greatly in the early 2000s. And so it's in a rebuilding phase for this top predator of the St. Lawrence River. And you can see a young juvenile here. So we come across these young juveniles in our survey occasionally. They're actually very rare. We've only caught 20 of them out of over 150,000 fish. So only 20 of them. So they're very rare. So we get super excited when we find them. We've identified 15 new sites as part of our surveys as well. And so we're really grateful for the support from Parks Canada and funding from Muskies Canada to continue that work. Round gobies, as mentioned, are almost at every single one of our sites. We have some collaborations on the go with multiple universities, Clarkson University and UCAM as well, to look at habitat associations with those round gobies, looking at how they are vectors for contaminants to flow through the ecosystem, and also how wetlands present these potential refuges of for species from round goby invasion, based on some of those physical characteristics of wetlands in the St. Lawrence River. This is a tube-nosed goby. You can see those little tubes coming out of the front of its face. We started to document these on the Canadian side of the river in the Thousand Islands in 2017. And they've since moved all the way down the river to Cornwall as of last year. So they've spread an incredible amount of distance and very quickly through the river. It's somewhat similar to the round goby. They do have some dietary differences. So it's in a learning phase right now. And we're really thankful that we developed a baseline data set before the species moved in. So now we can look at potential changes that's happening as they move in. So we're collaborating on this research with Dr. Don Farrell's group at SUNY ESF, as well as Dr. Anthony Ricciardi at McGill University. Another one I want to highlight, this is Maitland Tower. So this is near Prescott and Brockville. along the river. This is a great collaboration where we set up with Philip Ling, the property owner, where we're visiting the site many, many times, and we're looking at helping form and improve the fish habitat at the site as well. So it's always amazing when you can do monitoring research, but when that translates to actual improvements in fish habitat, I think that's the ultimate goal. We also started a great new collaboration with the staff at the Ganawage Environment Protection Office, or KIPO. That's Tyler Moulton and Brandon Rice there. We had a couple of great survey trips and visits with them last summer. They have some wonderful wetlands in that area. And over half the northern pike that we documented in 2021 came from one of those wetlands. So in at least one case, it seems to be doing pretty well. And this is a great collaboration that we're excited about developing and working together and moving forward. All of the data I've talked to you about today is one large data set that's informing this ecosystem indicator project called the Great River Report. And there's a little information here. There's a website, riverreport.ca. This is a state of ecosystem report of the St. Lawrence River that's being developed through the St. Lawrence River in collaboration with a lot of different partners that are contributing data sets. So I encourage people to check that out. And there's some great new graphics and lots of different indicators, including fish, water quality, invertebrates. So it's a great source of information for the public as well. Thank you to the 2021 crew. We have a large crew that goes out, you can imagine, with all the components of fish surveys and vegetation, water quality, environmental DNA filtering. Typical crew size is 6 to 7 people. And during COVID, it's quite challenging to move all those people around. So we're hoping that this year things calm down a little bit. This project has provided training to over 50 different people and counting, including people at the River Institute, the MCA, lots of different volunteers. Again, I'm just encouraging people who are working in the St. Lawrence River and they're interested to connect about this. You're very welcome to do so. And we'd love to hear from you. We are intending to go out in this year to continue our field surveys, continue the collection of environmental DNA. The analysis of previous years in eDNA samples is ongoing right now. And yeah, we're just really keen to work with people. If you want more information about this project, we do have a project website. If you just Google River Institute FINS, you'll find it. On that website, we do have links to a series of story maps that we've created for this project that are really engaging, that have maps and videos and photos. So it's a really great way of communicating the results. And you can also follow us on Instagram. So the team during the field season posts lots of great photos there. So thank you, and just thank you to all of our funding support. We've had funding from the beginning from Ontario Power Generation that provides core funding for this project. Again, we are a nonprofit institute trying to cover 200 kilometers of river and counting. So it takes a lot of funding and effort to do this. So thank you to Ontario Power Generation, the Trottier Foundation, and all these other organizations as well. And to our great partners at the MCA's Environment Program and all of our staff. So thank you very much.